Thanks very much, Evan. I really appreciate the invitation to come and share with you how at Recursion Pharmaceuticals we're using images to really try to revolutionize drug discovery. I'm guessing this is my clicker right here. So I first off want to start off by saying drug discovery in 2017 just could be a whole lot better. This is a plot showing the number of drugs that we get as a country, as the US, for every $1 billion that we spend on research. And down and to the right is bad. So this is not a good trend. And what this shows is it's actually about 100 times more expensive these days to get a drug to market than it was in 1950, and that's inflation adjusted. It's $2 to $4 billion, depending on who you ask. At the same time, there are thousands of untreated diseases out there. These are pictures of a few dozen children that are close to us at Recursion that suffer from a number of different genetic diseases. And so I want to contrast that with the amazing progress that we've seen in technology recently. Here's a picture of our lab and some robotics equipment that we had installed recently. It was a little under $3 million, and that's something that's a pretty affordable investment, even for a small startup like us, to be able to generate lots of biological data. At the same time, Amazon and Google are offering really simple, really efficient access to their huge cloud infrastructure so that you can fire up 10,000 CPUs at a moment's notice. And at the same time, artificial intelligence has advanced a lot recently. We've been talking a lot about this. And here's just one example from a few years ago where researchers were able to train a set of algorithms to be able to add captions to images. So the image you see at the top left, researchers were able to train this algorithm where it could identify that's a woman throwing a frisbee in a park. And if you highlight the word frisbee, the algorithm can show you where in the image it's getting the idea that there's a frisbee there. So with all of that progress in technology, what's holding back progress in biology and disease treatments? And I think a big part of that is that there's a standard formula that's used in the industry. And that is that, come here, not working well enough. Okay, yeah, that sounds like it's probably a lot better. So there's a standard formula that's used in the industry where there's a really kind of initial focus on one particular disease. You get maybe 100 scientists and invest $100 million over 10 years of research to try to develop a single biological hypothesis that drives the experiments you do. And those experiments, you often are just asking a single question. You want to identify a drug that has a certain type of activity. So you have a single endpoint experiment, and then you take the one or two best and you hone those and really kind of bet the farm on one or two drugs that you hope can treat that disease. And so we ask the question at Recursion all the time, how can we do better? And the way that we go about that is using images like this. So this is an image of human cells that are grown in our lab that we image using a number of different channels. So we have the first channel that I'll show you here is illuminating the nuclei of the cells. And this is a particular chemical called Hirscht, which really incalcates very nicely into DNA, very specifically. And then it fluoresces at this very particular frequency of blue light. And so we're able to capture these nuclei very cleanly. We do that with a number of different cellular components. So here I'm showing endoplasmic reticulum. You might have heard about this in your high school biology class. This is one of the many organelles in our cells that does different kinds of protein processing. Mitochondria, sort of the energy-producing engine of cells. You can see them kind of spidering away from the nuclei. Golgi apparatus, another of these possibly easy to forget cellular organelles that you learned in high school biology. But this is another protein processing organelle. This is a different chemical that illuminates RNA. And we've got another one here that's illuminating actin. And actin is really the structure, the skeleton of cells. It helps cells develop their shape and be able to move kind of throughout the tissue. So we put these all together, we get a beautiful image like this. And beyond being beautiful, I just want to zoom in a little bit and show you just the richness of the information here. So this, to me, really kind of harkens back to images that I've seen of the Hubble Deep Space Telescope. There's just kind of so much out there, so much information, so much richness that you could capture if you can find a way to do that. So we have these beautiful, rich images, and the other amazing thing about them is we can capture 100,000 images like this per week using that pretty small robotics platform that I showed earlier, costing just about $0.25 a piece. So images are rich, they're fast, they're cheap. How can they be used to fix drug discovery? So an interesting note about genetic diseases in particular is that there are actually hundreds of genetic diseases that can be diagnosed very effectively by doctors just by looking at the facial structure of patients suffering from these diseases. So on the top you see a healthy little girl, on the bottom a little girl suffering from a particular disease called Cornelia de Lange syndrome. Similarly with human cells, we can grow cells, the healthy cells you see at the top, and we can break a particular gene, the gene that's known to cause this disease, Cornelia de Lange syndrome. And on the bottom, you have a picture now of what are these disease model cells. So what can we do with that? Well, so we have healthy cells and disease cells that we can grow in these large experiments where we're actually going to test thousands of drugs independently, physically, putting these drugs on the disease cells and trying to identify the drugs that can make the disease cells go back to looking like healthy cells. And that's the real gist of what we're doing with a pretty substantial amount of effort right now at Recursion, is generating lots of images to do this. We don't stop there. We actually have about a dozen biologists who follow up on these experiments and do additional experiments, for instance, testing whether these drugs are effective in animal models of the diseases. And this has been able to very quickly give us about 20 different treatments already, potential treatments for over a dozen different genetic diseases just in about the last year of doing these kinds of experiments. So what do we do to the standard formula in the way that I just described? And you can summarize it by saying instead of one disease, we're looking at hundreds of diseases. Instead of 10 years, we're taking months. We don't have any biological hypothesis until after we find a drug of interest. Instead of a single endpoint experiment, we're extracting thousands of features from every single cell to make these kinds of decisions and analyses. And then instead of betting on one or two candidates, we're betting on 100 candidates for 100 different diseases. So this is already a massive improvement to the system and the problems that I was describing. But it's actually not where we stop. So what is our goal? What is our quest? And I'll describe what we see as kind of our 10-year quest. So we have, I think, a unique perspective at Recursion on what the field of drug discovery looks like. And I've tried to illustrate that here, where we have lots of diseases on the left, kind of symbolizing rows of a matrix, lots of drugs symbolizing columns of a matrix. And this whole space of potential intersections between diseases and drugs is the space of possible treatments. And so kind of taking that framing, if you look at our approach of testing individually, separately, physically, every drug against every disease, we sort of disparagingly call this our brute force search approach. And this is what we're doing right now, and it's really effective because images are so cheap and fast and rich. But what we imagine doing draws on some really recent advances in machine learning approaches called deep learning, convolutional neural networks that you probably saw some about yesterday. So this I'm showing right here is the flow of some of our cellular images through a convolutional neural network. And as you get to deeper layers in the network towards the right, you get more abstract and more composite features. Until you get to the end, you're extracting features from these images that are sort of the cellular equivalent of a face or a cat or a frisbee, like I showed you at the beginning. And so what does that allow us to do? So as I mentioned, our brute force search approach, we're really sort of testing drugs physically against diseases. And in doing that, we're capturing hundreds of millions of images. Meanwhile, biologists have generated massive amounts of biological knowledge about genes and how they relate to each other. Chemists have generated mountains of knowledge about chemical structures and how chemicals behave in different contexts. But they haven't had a bridge. They haven't had a way of putting all this together. And they haven't had a way of joining it with a large, highly structured, highly annotated data set. So that's where we come in. We use these advanced machine learning algorithms, deep learning, convolutional networks, various combinations of deep learning approaches to put together the existing biological knowledge with the chemical knowledge and with our big brute force search approach to generating massive amounts of images. And what we can start to do is predict which drugs should work for which diseases. So we might have seen this drug before, we might have seen this disease before, we've never seen them together before, but we can predict that this drug might work for this disease. Another thing that it allows us to do, going beyond that, is taking drugs that we've never seen before, predicting that they might work for diseases we've never seen before. So this doesn't seem that hard to believe that we can potentially do this in the future. This is actually a pretty reasonable projection. But what's kind of astounding is this biologists would call, I don't know if people here are familiar with the term AI complete to describe really difficult AI problems. Biologists might call this a biology complete problem. You really have to solve biology in order to be able to do this. So at Recursion, we are generating 100 million cellular images. We've got biologists and pharmaceutical experts together. We're putting it together with cutting-edge machine learning, doing all that to identify 100 treatments for 100 diseases in 10 years. And on top of that, our quest is to solve cellular biology. So that's Recursion, thanks to the power of images. Thanks.